The overall goal of this experiment is to evaluate if thyme essential oil applied in vapor phase can serve as effective preservative during meat drying process.
This method can help answer questions in the field of food processing and safety, such as how to effectively apply natural preservatives instead of synthetic ones. The main advantages of this technique is that it enables fast and equal distribution of thyme essential oil while the meat dries, and that all vapor can be used in lower volumes than in case of liquid oil application.
The use of this method can provide insight into improving the safety and quality of dried meat, and also can be applied for treatment of other food products against other unfavorable microorganisms.
Generally, individuals new to this method will struggle because the drop plate method used to count the number of the bacteria requires properly prepared plates and practice.
Obtain a short loin of beef from a local butchery and transfer it to the lab in a sealed bag at ambient temperatures within 20 minutes of purchase. In a laminar safety cabinet, sterilize the outer surface of the beef muscle using a 10-second application of 70% ethanol sprayed on from a squeeze bottle. Then, using sterile technique, cut out the outer surface of the muscle so that no ethanol remains on the preparation. Then, pack the muscle into a sealed bag and freeze it for 1 day at negative